In this video, we walk through the OligoWorks SPE workflow, providing context around each step in the protocol, along with guidance on recovery optimization and best practices. Let's begin with an overview of the OligoWorks SPE workflow, which is divided into two main steps. The first being the critically important sample pre-treatment step, and the second being the solid phase extraction, or SPE step. The primary goal of the sample pre-treatment step is to disrupt the tight binding that naturally occurs between endogenous proteins and oligonucleotides in biological samples, as this is critical to achieving maximum oligonucleotide recovery. In the OligoWorks sample pre-treatment protocol, we accomplish this by enzymatically digesting the protein in each sample using water's Rapizyme proteinase K, a highly active proteolytic enzyme, along with two other reagents that serve to enhance digestion efficiency. During the digestion process, proteins in the sample are cut up into small peptide fragments, and oligonucleotides previously bound are freed as a result, making them more accessible for capture in the following solid phase extraction step. Following the sample pre-treatment step, samples are then transferred to the SPE step and loaded into an OligoWorks SPE microplate. During the loading step, oligonucleotides in each sample selectively bind to the OligoWorks SPE wax sorbent through both ionic and hydrophobic interactions. After loading, sample wells are then washed twice using two different wash solutions that are designed to effectively remove the digested protein and other biofluid components contained in the sample. And lastly, captured oligonucleotides are then released and eluted from the sorbent using just 50 microliters of OligoWorks SPE eluent, producing a concentrated, low-volume eluate that's ready for direct LC-MS analysis. This is a key benefit of the OligoWorks SPE workflow when compared to detergent-based methods, which require evaporation and reconstitution prior to LC-MS analysis, lengthening the overall method time and adding increased variability as well. Now, let's look closer at the sample pre-treatment protocol, for which there are two choices. One, the standard protocol intended for biofluids, such as plasma or urine, and two, the solvent-assisted tissue homogenization protocol intended for tissue and/or cell-based samples. The standard sample pre-treatment protocol for biofluids shown here calls for the addition of 3 digestion reagents to the sample. 1. Guanidine hydrochloride, a protein denaturant. 2. TSEP, a reducing reagent. And 3. Rapizyme proteinase K, a highly active proteolytic enzyme, all of which are provided in the Rapizyme proteinase K digestion module included in every OligoWorks SPE kit and sold separately as well. The volume of each reagent added is directly proportional to the starting sample volume, and as shown, to a 100 microliter plasma sample, 20 microliters of guanidine hydrochloride, 10 microliters of TSEP, and 50 microliters of Rapizyme proteinase K are added for a total sample volume of 180 microliters. Note, to scale to different sample amounts, a volume guidance chart is included in the OligoWorks SPE Care and Use Manual. The reagent mixture is then incubated on a heated shaker at 55 degrees Celsius for 40 to 60 minutes at 600 RPM, depending on how much protein is present in the sample. For tissue or cell-based samples, our solvent-assisted tissue homogenization protocol has been developed to facilitate tissue homogenization and protein digestion in a single step. The same 3 digestion reagents included in the Rapizyme proteinase K digestion module are again used, but this time with added tris buffer and 30% acetonitrile to facilitate bead-based tissue homogenization first. This is done by adding all the sample and reagents into 2 milliliter Precelis tubes preloaded with ceramic beads, and placing them into a Precelis homogenizer, both from Burton Technologies. To homogenize, we recommend applying a 15-second pulse, waiting for 30 seconds, and then apply another 15-second pulse. The homogenized sample tubes are then incubated on a heated shaker at 55 degrees Celsius for 2 hours at 600 RPM. Following the incubation step, the homogenized, fully digested sample is spun down at 9400 RCF for 30 minutes, and an aliquot of the supernatant, typically 100 microliters, is then transferred to the following SPE step. Note, to learn more about the solvent-assisted tissue homogenization protocol, visit waters.com/oligoworks and click on the Resources tab where you'll find relevant application notes. Following completion of the sample pre-treatment step, samples are moved to the second half of the OligoWorks SPE workflow, the solid phase extraction step, where the load, wash, and elute procedures are the same for all pre-treated samples. The SPE procedure begins by positioning a waste reservoir in the lower half of a 96-well vacuum, or positive pressure filtration manifold, then stacking a gasket manifold assembly on top of the waste reservoir, and finally positioning the OligoWorks SPE microplate on top of the gasket manifold. In this video, we're using the Waters manual vacuum manifold, but other options can be used, like our semi-automated positive pressure system, the Auto Specialist, or our fully automated Extraction Plus vacuum manifold that integrates with our Andrew Plus pipetting robot and can operate independently as well. During the load procedure, oligonucleotides selectively bind to the OligoWorks SPE sorbent via ionic and hydrophobic interactions, and they remain bound as the digested protein and other matrix components contained in the sample are washed away in the following wash steps. For the standard protocol, the entire pre-treated sample volume, 180 microliters, is loaded into specified wells of an OligoWorks SPE microplate. Once all samples are loaded into the SPE plate, a slow, stepwise increase in vacuum or positive pressure is recommended, as described in the OligoWorks SPE Care and Use Manual, allowing residence time for oligonucleotides in the sample to adsorb to the OligoWorks SPE sorbent. Following the sample loading procedure, 2 wash steps are carried out. The first with 200 microliters of wash solution 1, an aqueous sodium acetate buffer, followed by 200 microliters of wash solution 2, an organic wash solution containing 30% methanol. With each wash step, the wash solution is added to sample wells in the microplate, and the vacuum or positive pressure system is slowly ramped up with the wash effluent flowing into the waste reservoir below. Following the 2 wash steps, we come to the final elution steps, where the goal is to release and recover the SPE-captured oligonucleotides for subsequent LC-MS analysis. Before implementing the elution step, the waste reservoir beneath the OligoWorks SPE microplate must be replaced with a Quan Recovery capture plate that has low binding properties and will capture the eluate from each sample individually. With the Quan Recovery plate in place, the final elution steps can be implemented, whereby 25 microliters of OligoWorks SPE eluent is added into each sample well and subsequently pulled down into the Quan Recovery plate below by vacuum or positive pressure, again using a slow stepwise increase in pressure as referenced above. After collecting the first 25 microliters of eluate, a second 25 microliter elution step is implemented, thus yielding 50 microliters of eluate per sample that can be directly injected into LC-MS systems for quantitative analysis. Having now reviewed the OligoWorks SPE workflow and protocol steps, the remainder of this video provides guidance on specific method adjustments that can be made to enhance recovery and/or lower matrix effects, as well as key best practices that can facilitate and accelerate new method development. Oligonucleotide therapeutics, including antisense oligonucleotides, ASOs, small interfering RNA, siRNA, and others, represent a highly diverse class of drugs in terms of their size, structure, and chemical makeup. So it's not surprising their binding affinities and extraction efficiencies can vary considerably. Should recoveries be too low and/or matrix effects too high when using the starting protocol, recoveries can often be greatly improved and/or matrix effects reduced through targeted method adjustments, and by utilizing some key best practices that we'll review now. Guidance on recovery optimization. First, let's review specific method adjustments that can help improve low recoveries and/or reduce matrix effects. Optimizing the standard pre-treatment protocol. Extend the digestion time. For most plasma or urine samples, a 40-minute incubation on a heated shaker with Rapizyme proteinase K results in complete protein digestion, as shown in this graph. But samples can vary considerably, and some may have higher protein content and require a longer digestion time. If oligonucleotide recovery losses are occurring during the pre-treatment step, it is likely due to protein in the sample not being fully digested. Should this occur, we recommend lengthening the digestion time to 50 or 60 minutes. Or, alternatively, increasing the amount of Rapizyme proteinase K that's added. Optimizing the tissue homogenization protocol. For lipophilic oligonucleotides, adding a small amount of NP40 detergent to the homogenization mix boosts recovery. Lipid-conjugated oligonucleotides are among the most challenging to recover, but our applications team, through their ongoing work, have shown adding a small amount of an eco-friendly, non-ionic NP40 detergent to the homogenization mix can significantly improve recovery of lipid-conjugated oligonucleotides in particular. Note, to learn more, see application note number 720008656 improved bioanalytical extraction of therapeutic antisense oligonucleotides, including a lipid-conjugated ASO from liver and brain tissues. Optimizing the SPE protocol. Dilute pretreated sample 1:1 with WASH-1 buffer before loading. Especially for polar oligonucleotides, a 1:1 dilution of the digested sample with WASH-1 buffer prior to loading can improve oligonucleotide capture efficiency, as doing so reduces the concentration of the guanidine hydrochloride, which can compete for sorbent binding via ionic interaction. Adjust WASH-2 organic composition. The standard WASH-2 organic composition contains 30% methanol. If looking to improve recovery or reduce matrix effects, a good place to start is with the WASH-2 solution. We recommend varying the percent organic from 10 to 50% and trying acetonitrile as an alternative organic to see if these changes have a beneficial impact. Adjust total wash volume and/or number of steps. If matrix effects are high, or if using NP40 detergent as part of your tissue homogenization pretreatment protocol, increasing the total wash volume and/or number of wash steps can help reduce matrix effects and remove excess detergent. Adjust total elution volume and/or number of steps. If recovery is lower than desired and other adjustments have already been tried, doubling the elution volume to 100 microliters and/or number of steps is worth a try. Dilute eluate 1:1 with water prior to LCMS analysis. To reduce the organic concentration of the eluate to better match starting reversed phase ion pairing conditions, a 1:1 dilution of the eluate with highly purified water prior to LCMS analysis has been shown to help and can potentially reduce matrix interferences as well. Key best practices. Use analytical reference standards to benchmark performance. Create control samples with one or more analytical reference standards spiked in, and choose standards for which extraction and LCMS performance are known. Note, Waters offers a range of oligonucleotide standards, including our MassPrep Oligo-DT ladder 15 to 35 μr, as well as ASO, siRNA, and single-guide RNA standards. To learn more, visit waters.com/GTX-standards. Spike in analytical standards pre- and post-sample pretreatment, as well as post-SPE, and collect the effluent from load and wash steps during method development in order to analyze and determine where oligonucleotide losses may be occurring. Calculate matrix effects using precursor and targeted MRM transitions. For better accuracy, be sure post-SPE spiked control samples are added to blank biomatrix that has been processed through the pretreatment and SPE steps as opposed to neat solution, as this will provide for a true apples-to-apples comparison. Use low adsorption sample prep and chromatography consumables. Oligonucleotides, being polyanionic, readily absorb to plastic, glass, and metal surfaces. And this nonspecific adsorption can cause significant losses in recovery, as well as increased variability. Shown here are two examples of the impact low adsorption consumables can have. The first showing significant loss in signal using a standard polypropylene capture plate versus our Quan Recovery capture plate. And the second showing a 10-fold increase in sensitivity when using our MaxPeak Premier columns versus standard stainless steel columns. Please visit waters.com/oligoworks for the latest resources. And please do not hesitate to reach out to your Waters representative or to our technical support team if you have any questions or need any additional information or assistance. Thank you. Waters, solving problems that matter.